We start chapter 3 of the A2 syllabus. The A2 syllabus is of course made up of 8 chapters, and the first chapter is respiration, and the second one is photosynthesis, and the third one is homeostasis. This is the syllabus biology 9700, and this is for the CIE, and we're going to study how homeostasis is maintained and how the body maintains homeostasis. Homeostasis means maintenance of a constant internal environment. And the first thing that we're going to study is how the three factors which are important for homeostasis, how are they maintained. The three factors which have to be studied is number one, thermoregulation. Thermoregulation means maintaining your temperature, the human body temperature, which must be maintained at 37 degrees Celsius. Then osmoregulation, which is of course the water content of blood. And as we know, the water content of blood means the water content of the plasma. Why? Because the plasma is going to be then turned into tissue fluid, and the tissue fluid bathes the body cells. So if we look at the body cells, we have capillaries, and till the fluid is inside the capillary it is called plasma. But once this fluid comes out, then it is called tissue fluid, and it surrounds the body cells. So the tissue fluid is very important, that the concentration of the tissue fluid must be maintained, because inside the cell cytoplasm has a certain number of solutes in it: sodium, potassium, chloride, glucose. So what we need to understand is that if the plasma, I'm just giving you an example, is a 2% solution, and the tissue fluid is also a 2% solution, and the cell cytoplasm is also a 2% solution, so then of course everything will remain intact. But if the tissue fluid becomes dilute, then what is going to happen? What is going to enter the cell? The cell is going to swell up and burst. Let's look at another situation in which the plasma becomes too concentrated, so it is 3%. So the tissue fluid becomes 3% instead of 2%. If it becomes 3%, we know the cytoplasm is 2%, so water is going to move out and the cell is going to shrink. Now we don't want that to happen. That would be disastrous. So number one, thermoregulation, number two, osmoregulation, and number three, we have to somehow maintain the blood glucose level. And I said blood glucose level, not blood sugar. And that has to be monitored, because basically glucose is the molecule which has to be respired to release energy. So it is the fuel. Just like in your car you need a fuel, you need either your car runs on petrol or it runs on diesel. So similarly your body works on glucose. Glucose is your body fuel. So it has to respire and energy has to be released. Now the first heading that we talk about is excretion in homeostasis, and that is the removal of toxic metabolic waste. Now the most important toxic metabolic waste, there are two of course that we talk about. Number one, we talk of carbon dioxide. Now carbon dioxide has been produced as a process by the process of respiration, and carbon dioxide cannot accumulate in the blood, so it must be removed, because it would be dangerous for too much carbon dioxide accumulating in the blood. So the lungs are going to remove the carbon dioxide from the blood and remove it from the body. The second very important toxic metabolic waste is urea. Now we have to understand first why and how is urea produced. Now, when you eat a diet which is rich in proteins, like you have meat or fish or eggs, the protein is digested to amino acids. The protein is digested to amino acids. Amino acids enter your blood and they are carried to your body cells. And the body uses them. Now the amino acids, the protein in your diet, in your food, is digested to amino acids. Amino acids enter the blood, is carried to the body cells, and it is used to make enzymes, to make hormones, to make structural proteins. But here comes the important part, which is that excess amino acids cannot be stored. Excess amino acids cannot be stored. So if they cannot be stored, then what do we do with them? So they cannot be stored. So what do we need to do with them? Either we can get rid of them, but if we get rid of them, they still have a lot of energy in it. So what has to happen is that they have to be deaminated. Now this is a process of deamination. So we say deamination occurs. Now where does the deamination occur? It occurs in the liver. So in the cells of the liver, the enzymes are present which are going to cause these amino acids to be deaminated. And we have to understand this process of deamination. Now deamination takes place in the liver, and if you look at it, there's a diagram of an amino acid. Now the part which I have drawn in red is the amino group. Now this is removed and a hydrogen added, and it is converted to ammonia. Now the remaining part is a keto acid, and it can either enter directly into the Krebs cycle, because please remember this is a 2 carbon, so it can enter as acetyl coenzyme A, or it may be converted to glucose. This is one of the possibilities. The other possibility is it can be converted to glucose. So we would need a couple of them to make a glucose molecule. Or it can be converted to glycogen, or it can be converted to fat. So this remaining part can either enter the Krebs cycle, or we can convert it into glucose and we can convert it into glycogen, or we can convert it into fat. But this ammonia part combines with carbon dioxide and it is going to form urea. Now this urea, this whole thing is taking place in the liver cell. As I said, liver. So this is taking place in the liver cell, and then this is going to enter the blood, and it is going to be carried in the blood plasma. So deamination takes place in the liver cells. The excess amino acids. The important thing that we have to understand is that excess amino acids cannot be stored. Excess amino acids cannot be stored. So as they cannot be stored, so they have a lot of energy, so we're going to remove the amino part, convert it to ammonia. Then the ammonia combines with carbon dioxide and we form urea. Now this whole thing is urea is occurring in the liver cell. And then this urea is going to enter the blood and is going to be carried in the plasma. Now the urea which was formed in the liver entered the blood and is carried all around the body. But when this blood circulates through the kidney removes the urea from the blood. Please understand this. The kidney removes the urea from the blood. And in order to understand that, we need to understand the structure of the kidney. Now, if you look at the kidney, Number one, you have a renal artery. This is a branch of the aorta. So the aorta going down is going to be somewhere here. And this is going to be the renal artery coming out of it. So the renal artery is a branch of the aorta and enters the kidney. The renal artery will divide into arterioles and then capillaries, and then the capillaries will join to form venules, and we have the renal vein coming like this. So renal artery going like this, which is a branch of the aorta, and the renal vein coming here, the renal vein coming out and entering the inferior vena cava. So what we have to understand is that the deoxygenated blood which is coming out of the kidney is going to enter the vena cava and is going to be taken up and will enter the right atrium. So we need to understand is that there are two blood vessels, one renal artery which is a branch of the aorta, and one which is the renal vein which is coming out of the kidney and will enter the vena cava, and the vena cava will take it back to the heart. But then there's another tube which comes out of it, and this other tube is called the ureter. So the ureter is the one which is going to carry the urine and is going to carry it to the bladder. There's a urinary bladder where the urine is stored, and then it's going to be passed out through the urethra. So we've got three vessels which come out of here. Number one, renal artery. Number two, renal vein. And number three, the one which is going to carry, this is called the ureter. So we have three tubes which are actually, if you cut a kidney section and you see three tubes, and you should be able to identify them. So the ureter is going to carry the urine, and that urine is going to go to the bladder. Now as we look at the kidney, we see a very interesting area. Number one, the outer area is called the cortex. The outer area is called the cortex, and the inner area is called the medulla. So you've got to know these two names. And then there is this area which is called the renal pelvis, and these areas which are these shaped like a pyramid is called the renal pyramid. And the basic unit of every kidney is a nephron. I will do more details of the nephron very soon. So this is the nephron, which is, of course, every kidney has millions of these nephrons, and it's a basic unit. That is why a doctor who deals with diseases of this is called a nephrologist. So the nephron is the basic unit, and as we proceed we will be doing more details of the nephron to see how it is placed and where it is placed in the kidney. So please pause here and go through these points again, and make a diagram and draw it in your copies and label it so that you remember all these. First we need to understand the structure of a nephron and the basic beginning of it. Now the basic beginning of it is that the renal artery enters the kidney, and as you can see, as it enters the kidney, it's going to divide into a number of branches, and each branch ends up in a little round structure which we call the glomerulus. Now you can see there's an artery which is going towards it, and there's an artery which is leaving it. Now we don't call it an artery, we call it an arteriole. And this glomerulus is made up of capillaries. So let's now try to understand this. So what we're going to have is, we're going to have the blood vessel which is going towards this glomerulus. So this is the one which is going towards it. Now this will be called the afferent arteriole. Afferent arteriole. and then of course is going to end up in these capillaries. And you know the capillaries have, so that's why I'm drawing a dotted line. So these capillaries are going to form a bunch of these capillaries here, which I might not have drawn very exactly. And then there's a vessel which leaves it. Now please understand, the vessel which leaves it is narrower. You can see the lumen of it. I've drawn this wider and I've drawn this narrower. There's a reason for it. That's how you find it in your body. Now this one which is leaving it, so E for exit. E for exit. But I don't want you to remember its exit. I want you to remember it as the efferent arteriole. Now if you look at it, this one is called the efferent arteriole, the one which is going out of the... So afferent arteriole is bringing blood to the glomerular capillaries, and this whole thing is called the glomerular capillaries. And this one which is leaving it, the one which is going to be coming out and going, the blood is moving out of it. Of course it has lost all the blood because it's all been filtered out here. This is called the efferent arteriole. And how are you going to remember it? E for efferent, E for exit. So efferent arteriole. Now if you remember, the normal pattern that we see in the normal pattern that we saw was what? There's an arteriole, there's an artery first of all. And then it's got branches, and these are called arterioles. And then there are capillaries. And then there is venule. And then there is a vein. So the normal is artery, arteriole, then capillary, then venule, and then vein. But here we see something very strange. We see afferent arteriole, and then we see capillaries. So these are the glomerular capillaries. And then you see an efferent arteriole. So arteriole, capillaries, and again an arteriole. And please remember, these are all found in the cortex. Cortex, if you remember, is the outer part of the kidney. So the outer part of the kidney is the cortex. So if you cut a section off and you see a microscopic examination of the cortex, well, you'll just see these glomeruluses. And you're going to just see a microscopic picture of that. So afferent arteriole, glomerular capillaries, efferent arteriole. And then as we proceed along it, we find that there is, as we look at it more closely, we see that these glomerular capillaries, there is a capsule around it, like a funnel shaped. And this capsule around it is called the Bowman's capsule. So this part is going to be called the Bowman's capsule. And what we have to understand is that this Bowman's capsule is actually to receive, because what is going to happen here is ultrafiltration. Ultrafiltration is going to take place here, and the blood is going to filter out into this, out of the capillaries. And this liquid here is called glomerular filtrate. So the liquid here is called, as it filters out, the liquid here is given a special name, and that is called glomerular filtrate. Glomerular filtrate. Now we go a little ahead and we say this is the afferent arteriole, this is the efferent arteriole. So A afferent, E efferent. Then this is the Bowman's capsule. And then we have the first convoluted tubule. Now this is called the PCT. PCT stands for proximal convoluted tubule. Proximal convoluted tubule, but it is accepted you can write PCT. So proximal convoluted tubule. Then I'm changing the color just to make you all understand it. Then there is a loop, and this loop is called the loop of Henle. This was scientists who discovered them. So loop of Henle. And then I'm changing the color again to show something. Now this is called the distal convoluted tubule. Distal, so this is also called the DCT, distal convoluted tubule. And this distal convoluted tubule opens up into another tube, which I've given another color to it. And this is called the collecting duct. Please pause it here, pause the video here, and let's look at it all once again. So afferent arteriole, glomerular capillaries, efferent arteriole, the one which is going to leave the glomerulus. Then we have the Bowman's capsule. This is the Bowman's capsule. Then the PCT, which is called the proximal convoluted tubule. And then we have the DCT. And in between these two, we have the loop of Henle. And the loop of Henle has a descending limb and an ascending limb. So it makes sense, descending limb and ascending limb. And then we have the distal convoluted tubule, which is distal or distant, and that opens into a tube which is called the collecting duct. So this entire structure is called a nephron. And what do we have? Another thing. This efferent arteriole is going to come back and is going to form a network around this. And this network is called peritubular capillaries. And then this network is going to form another set of capillaries around the loop of Henle. And those set of capillaries which are found around the loop of Henle have a special name which is called vasa recta. And then of course all these will join, and we will have the final, all these will join to form the renal vein. So this renal vein will be then leaving the kidney and carrying the deoxygenated blood away from the kidney. So once again, afferent arteriole, glomerulus, efferent arteriole, Bowman's capsule, PCT, DCT, loop of Henle. The efferent arteriole coming and forming a capillary network. So the efferent arteriole has coming back here and forming these, which are called the peritubular capillaries. The ones around the loop of Henle are called the vasa recta. And then of course they will join to form the renal vein, and the renal vein will leave the kidney. This is another diagram of and I want you to look at the labeling again. You can see here we have the afferent arteriole, then the efferent arteriole, then the glomerulus or the glomerular capillaries. Then we have the proximal convoluted tubule, You can see the peritubular capillaries, and then we have the loop of Henle, and you have the vasa recta surrounding it, and then we have the distal convoluted tubule, and of course this is the main tube which is going to carry the urine into the ureter and be carried it to the bladder. So this is again a diagram of the nephron, which I want you to visualize it in a little better manner so that you can now come up with all the labels and understand it. So afferent arteriole, efferent arteriole, glomerular capillaries, and then of course this efferent arteriole turns back. So this you can see here, the efferent arteriole going and forming the peritubular capillaries around the... Basically, what are the blood capillaries going to do? You see, the capillaries can't do anything. They're just like a river flowing. So what the capillaries are going to do is they're going to carry away the absorbed substances. Please understand this. Capillaries cannot do anything actively. It's just like a river, and you throw something into the river. So the river is just going to carry it away. So carry away the absorbed molecules. The glucose, the amino acids. So the absorbed molecules have to be carried away by the blood. So the capillary function is only to carry away the absorbed substances, like water is reabsorbed, glucose is reabsorbed, amino acids are reabsorbed. So all that is reabsorbed. Basically what we want to get rid of is, we only want to get rid of the urea. But we don't want to remove anything else from the blood. The urea has to be removed, the excess water has to be removed, and the excess salts have to be removed. So the kidney functions only for these things, is to remove the urea. Of course, urea is not excess, it is a toxic metabolic waste, so it needs to be removed totally. Then the excess water, if you've drunk a lot of water and it's a cold day, well, so much of water is going to increase the volume of blood. So we don't want that to happen. So that excess water has to be removed and the excess salts have to be removed. Now this will go on to the video 2, which will explain the further details of this. I've just discussed the structure here in this video, and further details would be in the second video which follows this video. Thank you.